This video will cover the beginning of Chapter 1, Module 1. We'll identify the cells that comprise the nervous system and explain how we know that neurons are not a continuous network with one simply running into the next. So the two main cells of the nervous system are neurons, the main communicators of the nervous system, and glia. We originally thought that glia just were the glue that held the brain together, and we know now there's so many other functions. And we'll discuss a little bit more about neurons and glia in subsequent videos. As you saw in the last video, the human brain contains approximately 86 billion neurons, once thought to be 100 billion, and somewhere down the road, new research might shed light on a different number. If you look at the different parts of the nervous system, the brain has about 12 to 15 billion neurons, the spinal cord about 1 billion, and the cerebellum has more neurons than the rest of the nervous system combined at about 70 billion neurons. And we'll come back and talk about different parts of the nervous system, different parts of the brain in later chapters. So now to the question of how do we know that neurons are separate, distinct entities? And we'll talk about the different parts of a neuron in a later video. Cajal, who dreamed to be an artist, taught us a lot about the nervous system. His drawings are these beautiful masterpieces that advanced the field of neuroscience immensely because we didn't have the technology that could do it on its own. So he wanted to be an artist. His father, on the other hand, wanted him to be a doctor and persuaded him to go to medical school. So he then was basically an artist trained as a scientist, and he used both of those talents to teach us so much about the field of neuroscience and the brain. So these are just a couple of drawings that he did, and so he would just spend hours looking at a microscope and drawing what he saw. Now, of course, today, we can just look at a microscope and snap a picture. We didn't have that technology then. So having all of these drawings allowed scientists to do things that they couldn't have done previously, and again, advance the field of neuroscience immensely. Again, these are hand drawings that he did. Here's of Purkinje cells in the cerebellum, and here's of the cerebellar cortex. Again, remember, that cerebellum has 70 billion neurons, more than the rest of the nervous system combined. So the other amazing things that he did was he used what's called a Golgi stain to stain these neurons. By doing this, he demonstrated that neurons remain separate. And I put neurons in quote because neither Cajal or Golgi is the person that coined the term neuron. That came years later. So in 1906, Cajal and Golgi won the Nobel Prize for determining that the neurons weren't this network that just continued on in the brain, that they were a separate and distinct entity. One started and stopped, and another one started and stopped. And what he did was he took sections of embryonic tissue and he stained them with Golgi's stain. It would look something like this, and he would take these thick sections and kind of go up and down, and he could tell that there was a separation, that the two neurons didn't run in together, and that it was too small of a gap to be seen by the microscopes of the day. Today, in order to see that gap, we have to use an electron microscope, and still all we really see is a fuzzy little line.